Olfactory membrane and receptor cell physiology of olfaction. Smell is the least understood of our senses. This results partially from the fact that the senses of smell are subject to phenomenon that cannot be studied with ease in lower animals. It helps to track food, can alert us to danger like gas leak, rotten food. It is also linked to brain that process emotion and memory. Olfaction or olfactory perception, the sense of smell mediated by a group of specialized sensory cells in nasal cavity, order the property of a substance which gives a particular smell. Olfactory membrane lies in the superior part of each nostril. In each nostril, the olfactory membrane has a surface area of about 2.4 square centimeters. Olfactory cells, the receptor cells for the smell sensation, bipolar nerve cells derived originally from the females, from 100 million of the cells in the olfactory epithelium interspaced among sustentacular cells. And composition of olfactory epithelium. Each neuron has a thick dendrite with an expanded end called olfactory road. From root, cilia project to the mucous surface. Each receptor neuron has 10 to 20 cilia. Axons of olfactory receptor neurons pierce cribriform plate of ethmoid bone and enter olfactory bulbs. So olfactory neurons have half time of few weeks. Next, mucus producing glands. Olfactory mucous membrane is constantly covered by mucus. Mucus is produced by woman's glands placed just beneath the basal lamina of the membrane. Then olfactory bulbs. The olfactory bulbs relay sensory signals to the olfactory tract. Small axons from the olfactory epithelium synapse with receptor neurons and interneurons in the olfactory bulbs. Olfactory membrane. The mucosal end of the olfactory cell forms a knob from which 4 to 25 olfactory hairs, also called olfactory cilia, project into the mucus that coats the inner surface of the nasal cavity. Mucosal end project into the mucus. Then cilia react to odors in the air and stimulate the olfactory cells. Spaced among the olfactory cells, Bowman's gland that secrete mucus into the surface of the olfactory membrane. Olfactory cells are constantly being replaced with a half time of a few weeks. So excitation of the olfactory cells, the proportion of each olfactory cell that responds to the olfactory chemical stimuli is the olfactory cilia. The odorant substance, on coming in contact with the olfactory membrane surface, first diffuses into the mucus that covers the cilia. Then it binds with the receptor proteins in the membrane of each cilia or OBP. Then receptor protein, G protein, CAMP pathway, opening of sodium channels, action potential exciting the olfactory neuron and olfactory nerve CNS. Then G protein is stimulated, which triggers activation of adenylyl cyclase enzyme, speeds up the conversion of ATP to CAMP. CAMP then binds to action channels in membranes of cilia. This causes channels to open and C ions to enter cilia. Influx of C ions activates Cl minus channels to open and Cl leaves. Membrane becomes depolarized and AP is created. The action potential travels down the axon of olfactory receptor cell, eventually meets with another axon and forms the olfactory nerve. Olfactory tract, olfactory bulb, mitral cell, glomerulus, sustentacular cells, olfactory cilia, mucus layers, Bowman's gland. Then excitation of the olfactory cells, only volatile substances that can be sniffed into the nostrils can be smelled. Substances must be at least slightly water soluble so that it can pass through the mucus to reach the olfactory cilia. Substances to be at least slightly lipid soluble, presumably because lipid continents of the cilium itself are a weak barrier to non-lipid soluble odorants. Sniffing is a semi-reflex response that usually occurs when a new odor attracts attention. The olfactory receptors adapt about 50% in the first second or so after stimulation. Thereafter, they adapt very little and very slowly. Our own experience that smell sensations adapt almost to extinction within a minute or so after entering a strongly odorous atmosphere. Adaptation, because this psychological adaptation is far greater than the degree of adaptation of the receptors themselves. Most of the additional adaptation occur within the CNS. Larger number of nerve fibers pass from the olfactory region of the brain backward along the olfactory tract and terminate on special inhibitory cells in the olfactory bulb, the granule cells. Primary smell sensations: camphoraceous, musky, floral, pepperminty, eternal, pungent, putrid smells. So like that. 100 primary sensations of smell are there. Affective quality of either pleasantness or unpleasantness. Smell is probably even more important than taste for the selection of food. A person who has previously eaten food that disagreed with him or her is often nauseated by the smell of that same food on a second occasion. Then threshold for smell. The minute quantity of stimulating agent in the air can elicit a smell sensation. The substance methyl mercaptan can be smelled when only 1/25th trimillionth of a gram is present in each milliliter of air. Because of this very low threshold, the substance is mixed with natural gas to give the gas an odor that can be detected when even small amounts of gas leak from a cylinder. And it's often said that females are more capable of detecting the smell. Smell pathway. Olfactory bulb, olfactory tract, olfactory nerve. Olfactory bulb, olfactory tract, olfactory nerve, first radial nerve. Both the tract and the bulbs are an anterior outgrowth of brain tissue from the base of the brain. Olfactory bulb lies over the cribriform plate, separating the brain cavity from the upper nasal cavity. The cribriform plate has multiple small perforations through which an equal number of small nerves pass upward from the olfactory membrane in the nasal cavity to enter the olfactory bulb in the radial cavity. Ranium, cribriform plate of ethmoid, olfactory bulb, olfactory tract, olfactory mucous membrane, superior, middle and inferior turbinates. Superior, middle and inferior turbinates. Then hard palate. Smell pathway. Short axons from the olfactory cells terminating in multiple globular structures within the olfactory bulb called glomeruli. Short axons from the olfactory cells terminating in multiple globular structures within the olfactory bulb called the glomeruli. Each glomerulus is the terminus of dendrites from about 25 large mitral cells and about 60 smaller tufted cells, the cell bodies of which lie in the olfactory bulb superior to the glomeruli. Granule cells, periglomerular cells. Mitral and tufted cells send axons through the olfactory tract to transmit olfactory signals to higher levels in the CNS. So glomeruli, short axons, mitral cells, tufted cells superior to the glomeruli, granule cells, periglomerular cells. Then the two mitral and tufted cells send axons through the olfactory tract. Then mucous cilia, axons of olfactory cells, glomeruli in bulb, dendrites of mitral, tufted cells in bulb, axons of mitral, tufted cells in tract, CNS. Smell, CNS pathway. Olfactory tract divides into or mediates into the median olfactory area of the brainstem, very old olfactory system, either passing literally into the lateral olfactory area, stria, a newer and less old system, the medial olfactory area, very old, septal nuclei, hypothalamus, limbic system, removal, not much effect, the less old lateral olfactory area, prepariform and pyriform cortex plus portion of the amygdaloid nuclei, limbic system, hippocampus, learning, aversion. Then small CNS pathway, smell CNS pathway, lateral olfactory area, anterior medial portion of the temporal lobe, cerebral cortex. This is the only area of the entire cerebral cortex where sensory signals pass directly to the cortex without passing first through the thalamus. The newer pathway pass to the thalamus, passing to the dorsomedial thalamic nucleus or orbitofrontal cortex, conscious analysis of order, granule cells, periglomerular cells, lateral inhibition, hypothalamus, medial olfactory area, prefrontal cortex, olfactory tract, mitral cell, olfactory bulb, olfactory nerve, orbitofrontal cortex, lateral olfactory area, brainstem, hippocampus, then temporal cortex, etc. And frontal lobe, olfactory bulb with cut ends of olfactory nerves, olfactory tract, anterior commissure, temporal lobe. Note location of uncus relative to brainstem, then cut surface of brainstem, parahippocampal gyrus, anterior perforatory substance, renal sulcus, rhinal sulcus, and then uncinate gyrus, unkif, then orthofrontal cortex. Omeronasal organs in rodents and various other mammals. The nasal cavity opens. Nasal cavity contains another patch of olfactory mucous membrane located along the nasal septum in a well-developed vomeronasal organ. The structure is concerned with the perception of odors that act as pheromones. Its receptors project to the accessory olfactory bulb and from there primarily to areas in the amygdala and hypothalamus that are concerned with reproduction. The organ is not well developed in humans. Omeronasal organ evidence for the existence of pheromones in humans. Close relationship between smell and sexual function. The perfumats. The cells of smell is said to be more acute in women than in men, and in women the most acute at the time of ovulation. Smell and to a lesser extent taste have a unique ability to trigger long-term memories. Smell and to a lesser extent taste have a unique ability to trigger long-term memories. Pain fibers in the nose, naked endings of many trigeminal pain fibers are found in the olfactory mucous membrane, stimulated by irritating substances, peppermint, menthol, and chlorine, sneezing, lacrimation, respiratory inhibition, and anosmia, absence of the sense of smell, hyposmia, diminished olfactory sensitivity, and dysosmia, distorted sense of smell. Then olfactory thresholds increase with advancing age, and more than 75% of humans over the age of 80 have an impaired ability to identify smells. Then anosmia, and hypogonadism, Kallmann syndrome.